Good evening, everybody. Thank you so much for the kind introduction and the invitation to come here. I'm having a great time. You guys are the best hosts. Really enjoying getting to know you and this incredible university that was built just under 40 years ago. So today I want to talk to you about the work that was performed in my lab over the last 20 years. But before I start with the work, it's important for me to show the picture of all the people who actually did the work. So I'm a professor, and many of the people who actually do the work are PhD students and postdoctoral fellows. And this is from a recent lab reunion just a few months ago. So these are all the current lab members as well as alumni who came back from all over the world to celebrate science. The other way I like to show and acknowledge people is by this map, which I'm both proud of and a little bit of embarrassed about, and I'll tell you why. So this is obviously the map of the world, and each of these blue pins shows one of the people in my lab who came to my lab and was born in this area. So as Andrea said, this is me, born in Lebanon. And you know, we're doing very well with very good diversity from all over the northern hemisphere. But as you can see, we're not very well represented in the southern hemisphere. And hopefully that'll change, and I hope to have someone from Ecuador come to my lab in the near future. So your perception of reality is based on your senses, and over the last few decades, scientists have figured out how you can taste, smell, or see. However, highlighted in red here is this sense of touch is much more enigmatic. It gathers information of mechanical, thermal, or chemical nature and represents this in the brain, and how we sense this, specifically at the first step of how you transduce this physical stimuli into electrical signal that neurons understand, had not been known for quite a while. Just give you some introduction of how temperature and pressure is so important to us. We are very sensitive and particular about temperature. This is one example. So luxury cars about 20, 30 years ago realized that humans prefer very specific temperatures. As you know, probably you all argue at home of what temperature to set the thermostat. And car companies have realized that they can actually control the temperature for the driver and passenger separately. It's just a sign of how much we care about temperature. Another extreme example is wine. So these are both fermented grape juices. And as you know, red wine versus white wine, we prefer them to be stored and drank at very, very different temperatures. So the other side of this somatosensation is sensing touch. And I'm here to tell you that your sense of touch is quite amazing. So humans can discriminate indentations that are 1/500th of a diameter of a hair. That's quite remarkable. So you have incredible ability to sense touch. But in addition to this discriminative ability, we rely on affective touch for our emotional health. And we've all noticed this during isolation, during COVID period, for example. And the related sense of proprioception, which really tells us where our limbs are in space without looking at them. So this is how I can close my eyes and touch my nose. Or these very gifted musicians, Yo-Yo Ma and Emmanuel Axe, are playing instruments, very complex tasks, without even looking at their hands. But to me, a picture that shows the power of touch is this one. This little kid is in the White House Oval Office to meet President Obama, and he tells him that his friends tell him that his hair looks like the president. So the president immediately bends over and says, "Why don't you touch it?" And so I'm not just talking about the texture of the hair here, but once again, the powerful nature of affective touch. And many people say seeing is believing, but clearly touching is believing as well. So the flip side of touch is pain. And acute pain is absolutely essential for our survival. There's actually people who have mutations so that they don't sense pain at all, and this is extremely dangerous. These people think they're invincible. Some have known to jump from 5-story buildings and die just because they thought they just can't feel pain, so they can do anything. But chronic pain, which we define as pain that persists even when the stimulus that caused the pain is gone, is called chronic pain, is a major unmet medical need. So when I talk about temperature sensing, touch sensing, pain sensing, proprioception, I want to tell you how this all gets initiated. So this is a video of a zebrafish that's imaged. It's a time lapse, and what you can see is these little blobs are actually thousands of neurons in what we call a ganglia that send processes, little dendrites or axons, innervate the skin and many other parts of your body, where they sense this physical stimuli and translate it, send it to the spinal cord. So this is a video of a developing zebrafish, and these little processes again are going out everywhere, muscle, skin, internal organs, to collect information to send it to the spinal cord and the brain. So these neurons, of course, are microscopic. The cell body, as I said, is one of these ganglia, has thousands of neurons. You can't see it, you need a microscope. But one of the more fascinating things about them is that they're the longest cells in your body, because this one neuron sends one process to the skin, and the other end will go all the way to the brain. So if you hold your touch neuron, like Yao Ming is doing, one of the tallest basketball players who stands at over 2.3 meters, one of his single neurons is up to 2 meters long. That's quite phenomenal. So if we look at this cartoon of this neuron, again, their job is in a way quite simple. From one end, they sense these physical forces, translate it into electrical signal that then goes to the brain. But what we're really interested in is the receptor, the molecule, the DNA that codes a protein that will sense temperature. How do you sense temperature? How do you sense pressure? And of course, like many other functions in your body, everything almost is done by a protein. I know many of you know this very well, but this is a very specialized protein called an ion channel. But what is a protein? Almost everything that your body does relies on proteins. Hormones, insulin is a protein. Structural proteins, we call them keratin, in your nails and your hair, give you the structure of these tissues. But there's also lots of decision-making proteins that we call sensors, for example, and these are ion channels. The next slide will tell you what an ion channel does. So cells, each cell is covered by a lipid bilayer, and it's a very creative thing to do for a cell because it protects it from the rest of the environment. But it comes at a disadvantage because this lipid bilayer then can't talk to the outside of the cell, which is very important to do. So ion channels is one of these many proteins that are kind of signaling molecules that allow the cell to know what's going on outside. And ion channels, as the name suggests, they conduct ions. So they do either nothing, they're closed, or when they're open, ions flow into the cell. They're remarkable machines. Once they're open, 10 million ions can flow through them per second. And for neurons, once this flow occurs, it actually is enough ions that changes the membrane voltage of the plasma membrane. And this causes an electric impulse that travels from neuron to the brain. I won't get into this, but many drugs are targeting various kinds of ion channels, including for conditions such as anxiety, arrhythmia, cystic fibrosis, epilepsy, stroke, and also anesthetics that you use during surgery. But we're looking for a very specific ion channel that's not activated by chemicals, but again, activated by physical forces such as force or temperature. So how do you sense this gentle breeze or a cactus pricking your finger? Before again I go into one more slide of introduction, I want to give you an image of how small these ion channels are that we're trying to find. So this is a to scale a grain of rice. And these cells that I'm talking about, covered by plasma membranes, are 500 times smaller, so that represented by the tiny dot. So if we amplify this dot, this is what a cell schematic looks like. It's about 10 micrometers. And so the ion channel would sit in the plasma membrane, but it's so small that even at this scale, you don't see it. The smallest ion channels are 0.9 nanometers, can be up to 20 nanometers in diameter. So much, much smaller than a cell. I also want to tell you why is it important to identify these sensors. You know, in science, I call myself a basic scientist. What that means is, what drives me is curiosity. To me, it's very interesting to figure out how we have evolved to sense these physical forces. So how do you translate temperature or mechanical force into chemical signals? By itself is a very interesting question. But obviously, as I mentioned, finding these molecules and them being a target for future drugs could be an important application for treating pain. And one of the wonderful things about studying basic science is that sometimes you get applications that were completely unexpected from where you started, and we'll touch upon this in a second. So what I want to tell you today is very quickly two directions from my lab. We found these sensors for temperature, and I'll give you two quick examples of ion channels called TRPM8 and TRPA1, and also mechanosensors that we call PIEZO 1 and 2. In each of these stories, we start with a very reductionist system, identify the proteins or the ion channels, and then we move on into in vivo systems. So what are they doing in a whole animal? We use animal models such as mice or other species, but we also get data from clinical human data. So the first ion channel that my lab identified back in 2002 is a cold activated ion channel, and we call it TRPM8. So how do we know that this channel is activated by cold? This is one of the fascinating techniques that I want to share with you. It's called a patch clamp, where you electrically monitor the activity of this ion channel. So this schematic shows this little patch, glass patch, surrounding piece of a plasma membrane in blue, and there's one channel sitting in there. And this is actually an experiment where you cool this medium around this ion channel and record electric impulses. So what you're seeing here, this tiny flicker of red, is actually a single protein, those tiny little machines that I showed you, opening and closing. And if you keep cooling this pipette, you can see that there are actually 3 channels in this, because 1, 2, and 3 channels all start opening at the same time. Just to give you an idea, this y-axis shows that the amount of current is about 15 picoamp. So a 100-volt light bulb is about 1 amp. So the amount we're talking about, if I wrote this in amp value, would be 0.14015 amps. So this is very, very tiny. So we need very sensitive machines to be able to record this kind of electrical activity. One of the interesting findings from this was that this channel that's cold activated was actually also activated by menthol. Menthol is a natural product that's derived from mint leaves, and it's actually, I don't know if you have these brands in your pharmacies, but many over-the-counter products such as toothpaste, anti-pain rubs, etc., use menthol for its cooling sensation. And if you really want to find out a menthol product, how it's cooling, if you brush your teeth with one of these toothpaste and then drink slightly cold water, you'll get that very fresh, minty coolness. So it's fascinating that this plant has evolved a compound that activates the cold channel, and we actually think of it as cold. I'm not going to show you, but we've shown, as I've mentioned, in vivo, that these are actually absolutely required for cold sensation. So if we engineer a mouse that doesn't have this channel, it loses its ability to distinguish cold temperatures. Another ion channel we identified is called TRPA1, and instead of it being activated by cooling compounds, this channel is activated by many, many things that we consider spicy. So for example, I don't know, again, you have this brand, but there's mouthwashes that have this burning sensation in the mouth. And although they might include alcohol, which is the real antibacterial, they put these chemicals so that you feel this burning sensation. And humans, think, oh, it's burning, it must be killing bacteria. All it's doing is actually activating your TRPA1 channels. If you like sushi, again, every time you dip your soy sauce with a little bit of wasabi and have that pungent sensation in the mouth, it's again a TRPA1 activator. Even raw garlic has a chemical that activates TRPA1. So this sounds all fun and good, but actually the research that followed this is our understanding that TRPA1 is not just being activated by spicy compounds, but almost all reactive compounds that either from outside or inside we produce. So TRPA1 is a general sensor for chemical damage. And now work from our lab and later pharmaceutical companies have found blockers of TRPA1. And as we speak, these are actually in phase 2 clinical trials to see if these compounds are beneficial against various indications of pain. This is all I was going to say about the temperature side. Over 12 years ago, we decided we really needed to switch, since we found those receptors, and find out how do you sense touch, how do you sense mechanical force. And this is, Bertrand Coste was a postdoctoral fellow in my lab. He since then went back to Marseille, France, as a professor there. And he took a very, very, what we call a reductionist approach again to find a sensor for mechanical force. Once again, we find it in a cell, and then we go back into the animal and see what it does. So in this assay, this is a glass probe again, electrically recording from a cell, and this time, instead of changing the temperature, with another glass probe, he comes and pokes the cell. So if this cell is expressing this protein that we want to know about, then we can record this mechanically activated current shown here at the bottom. So the more you push the cell, the larger this inward current that we can record. So how do you go from this assay to actually finding the protein that senses touch? So first he recorded from 30 cell lines. These are easy to work with cells that we can grow in a dish in the laboratory. And he finally found one that has this robust activity. And then I won't go into much detail, but he identified, using bioinformatics and other tools, candidate molecules, potential proteins, genes that code these proteins, that could be ion channels. And the list was about 300. And then using another newer technique called RNAi, which is you can knock down the expression of one protein at a time, he would knock one down and see, is this mechanical activation still there or not? So this is the experiment that he did. Kind of became a famous experiment. So what you're looking here at the x-axis is this current, the about 100 picoamps, 200 picoamps that he can get from these cells when he pokes them. And then each of these little circles that you see is one of his candidates that he's knocking down and saying, is there still activity? Anything between these two lines means the activity is quite normal still. But think about it. To test each of these candidates, he had to spend 2 to 3 days full time knocking them down and recording. So Bertrand had 11 months of complete negative, unproductive work in the lab. So for a whole year, he just came in, did an experiment, went home. His wife asked him, did you find anything? He said, nope. But he persisted, and finally, the 72nd candidate that he tested was called unknown family 38A, which means we have the sequence, but we have no idea what it does. And so it was immediately clear to him that this was an important one, and he renamed it PIEZO1 from the Greek piezi, meaning pressure. So just to summarize, what we call now is that these piezos are both necessary and sufficient to induce mechanosensitivity. These are important words for scientists. Necessary means the previous experiment. This cell is mechanosensitive, you take it away, it's no longer mechanosensitive. Sufficient means you take this protein now, put it in a naive cell that's not mechanosensitive, and by itself is enough to make it mechanosensitive. So these are, if you have these two conditions, you say, we're onto something interesting. And then by just comparing the sequence, we know that there are at least 2 piezos in mammals, piezo 1 and 2, and I'll talk about this in a little bit. And they're not just in humans, they're actually in many animals, including plants, and I'll get to this at the end. Even pathogenic protozoa, single cell organisms have piezos. More fascinating is that they have a structure that's very, very unique. So these ion channels that I told you about, for them to sit in the membrane, they have to have what we call transmembrane domains, a piece of protein that can go through the fatty lipid bilayer. And most ion channels have 2 to 6 of these. We found out that piezos have 38. So that's more than any other protein in the genome. On top of that, we solved the structure by using cryo-electron microscopy. And we found that 3 of these proteins come together to form the functional ion channel. So this is actually what the channel looks like. I told you 3 of them come together. Each is shown in a different color. So at the center where the 3 meet is the pore where the ions would go through. The rest, each of these ribbons that you see, is one of these transmembrane domains, and they kind of extend out into the plasma membrane like antennas, trying to sense any tension in the lipid bilayer. So what we imagine they look like, if blue is this lipid bilayer of a cell, these proteins kind of dig a hole and sit like that in there. And any stretch of the membrane, they're supposed to sense touch or pressure. They we think they flatten to activate the ion channel. And actually, one of my trainees solved the structure of this flat protein, and this is morphing of the 2 channels, closed and open. I show it because I find it so beautiful. It's like almost an engineer would be very interested in this, because it's a molecular machine. So you go from closed, you stretch it, and the whole protein moves and stretches and becomes flat, allowing ions to go through. I'll show you one more time. Okay, now we've kind of characterized this ion channel as a pressure sensitive, necessary and sufficient. As I promised, we really want to know what happens in an animal when we don't have piezo. And the first animal we used, model system, is mice. There's lots of good genetics in it, so we delete the piezo gene in mice and ask if they still sense touch. We have done many quantitative experiments, but what I want to show you videos of the first experiment we do, which is very not sophisticated, but it's kind of fun to look at. So this is what we call a wild-type mouse. A normal mouse, all their genes are as they should be, and we call this assay tape on the back. We put a white piece of tape on the back and see what they do. So as you can see, they don't like it and they try to take it off. And here's an animal that doesn't have a functional piezo 2 gene. They're completely oblivious. Again, I'm not going to show you many quantitation, but we have a pretty good idea that in mice without piezo 2, these animals don't sense touch. How about proprioception? I love talking about proprioception because I think it's your most important sense, but you don't even probably know about it. So proprioception is the same types of neurons that innervate the skin for sense of touch, but these neurons innervate the muscle. You're not sensing your muscle being stretched, but from this information, you learn where your limbs are. As I told you in the beginning, this is how else would I be able to close my eyes and touch my nose? I have a sense of how much the muscles are stretched, and from that combined information, that's why when you're first born, you don't have very good proprioception, you can't walk, you don't look coordinated, but with time, you learn this and can do very coordinated things, just walking, or in this case, some very complex walking blindfold if you're talented and train a lot. So we showed in the mice that without piezo 2, proprioception is completely ablated. I won't show you that because I want to show it to you in humans. My collaborators at the National Institutes of Health, Alex Chesler and Karsten Bonneman, actually identified individuals who lack a functional piezo 2. So there's now at least 20 people who have this kind of condition. So they're complete ablated of piezo 2. And remarkably, they have the exact same phenotype as I've described to you in the mice. They don't sense touch at all. They don't have proprioception. And this video shows you what happens to individuals who don't have proprioception. So she's blindfolded and asked to walk in a straight line. You and I can do this very easily, but as you will see, she has a very, very hard time doing this. She's harnessed so she doesn't fall down. So how about pain? We've shown both in mice and in humans that acute pain is relatively normal without piezo 2. However, we're very interested in this condition of pain called tactile allodynia. So two complicated words, but a very simple idea. Tactile allodynia means when touch becomes painful. So you experience this after sunburn, for example, or injury, where just touching the area becomes painful. But this is much more important in the clinic because patients suffering from neuropathic pain have this condition all the time, and there's no good medications for them. So once again, we showed that in mice without piezo 2, there's no tactile allodynia, which is very good. It means that piezo 2 could be a good target for pain drug. But interestingly, again, our collaborator Alex Chesler, because there's these individuals who are deficient in piezo 2, we can directly, simply test this in humans. So what they do is, again, they're blindfolded, and you have two imaginary boxes on the arm. One is control, gets nothing. The other one gets capsaicin, this active ingredient in chili pepper that causes inflammation. And then you have a control group, and then those that are piezo 2 deficient, and you simply ask them, if I touch the inflamed part versus the other, do you feel pain? So in the control group shown in black, every single person knows exactly that B box is painful, not A, but the individuals that don't have piezo 2 can't sense pain at all. But you tell them the way this assay works, you have to guess A or B. They randomly guess, they get it right 50% of the time. So now we know that piezo 2 is actually not important for acute pain, but at least for some forms of chronic pain. So what I've told you till now is that both from studies in mice and humans, piezo 2 seems to be playing an important role in detecting mechanical force relevant for touch sensation, proprioception, some aspects of pain, as well as mechanical itch that I didn't talk about. Mechanical itch is when you wear a wool sweater and it itches like crazy, or a mosquito jumps on you and you have to detect it because it itches and get rid of it. This is also true piezos. But one of the interesting side stories that's developed over the years when we've collaborated with Steve Liberless at Harvard and Alex Chesler at NIH is that these same neurons also innervate many internal organs and play important role of mechanosensation there, although their role in internal organ sensation was very little described. So for example, every time you take a breath, these molecules sense how much your lung is stretched and control your breathing pattern. Baroreceptor is a mechanism of how you sense blood pressure and control it. So if you go for a run, your blood's going to pump much higher, but you don't want your blood pressure to go up. So these receptors sense the stretch of the aorta and control blood pressure, and you're completely unaware of this. So this is happening at a not conscious level. But you are very conscious when you have to go to the restroom because your bladder actually gets stretched, and that is what you're sensing. And again, both in humans and mice, this seems to be regulated by piezo 2. So that the humans without piezo 2, ever since they're kids, they've learned that they have accidents all the time. So they go to the restroom on a schedule not to have these accidents. But I want to end this section by just telling you that this is a very active area of research. So for example, one of the things we're looking at in the lab right now is another organ stretching, not bladder, but stomach. This is a very interesting cartoon showing how you feel when you're very full. And my hosts here at the USFQ have been feeding me a lot, so I've been getting this sensation a lot. So in biological research, I would say 95% of the research on satiety, which is fullness, is actually on chemicals. It's nutrient sensing. How much glucose, how much fat, how much protein, that sensed and tells the brain maybe you should stop eating. And all of this is of course very important. But as you and I know, when we eat a lot, the signal that we feel is that stretch of the stomach. But there's almost no understanding of how much this stretch contributes to how much you eat, when you stop, diabetes, obesity. There's nothing known about the contribution of the mechanical sensation to these processes. And these molecules give us this kind of new route to study it. As I told you, I love biology and discovery science, but I always get asked, how does this help patients? And I think, although our main goal is basic science, there's been many applications to this and growing ones coming up. So for example, clinically, when someone goes to the doctor's office and is very uncoordinated, what they immediately think that there's a motor neuron problem or a muscle problem. And so all of these 20 patients initially were misdiagnosed till genomic sequencing helped them identify that it's a mutation in PIEZO2, which then realized that, this is a sensory problem. So sensory problem is not on the forefront of most medical doctors, and this kind of genetic research has helped that already. But as I mentioned, both in mice and humans, we now know that blocking PIEZO2 might be very beneficial as a potential drug against some forms of pain. And we think a topical use of a future inhibitor that we and others are working on might be very useful for this. And also, some areas that I didn't think about when we first started this, but with respect to bladder dysfunction, hypertension, all of these are possible indications that piezos might be playing an important role. I'm going to take one step away from neuroscience and just say that it's not just sensory neurons that sense pressure. Many cells, blood vessels sense shear stress, muscles get stretched, red blood cells get indented as they squeeze through capillaries. And there's a whole lack of knowledge of how these types of cells sense mechanical force and how they respond to it. And many of the research on piezo from our lab and others are focusing on these other systems. For example, I won't talk about this, just to mention it, we know both from mouse genetics and human genetics, without piezo1, the sister molecule of piezo2, your lymphatic system doesn't form and you have lymphedema. Piezos are also playing an important role in bone formation. You might have heard that astronauts, when they go into outer space for a considerable amount of time, they come back with reduced bone density. And research in other labs, not mine, have shown that without piezo1, bones have much lower bone density, so this might be related. In addition, we and others have shown that piezos play a very important role in a variety of types of blood cells, including red blood cells and white blood cells. But I want to kind of end the talk by taking a step back and reminding you that piezos are not just in humans and mammals, but in many species. So piezo, this is we call it evolutionary tree. In the gray is the animal kingdom. Drosophila, fruit flies have it. C. elegans worms have it. Unicellular organisms have it. Even plants have it. And one of the postdoctoral fellows in my lab actually studied the role of piezos in plants. So plants experience mechanical force as well. For example, a plant root, when it's going through the soil and experiences a hard surface, it has to decide whether it's going to go right through or go around it. That's mechanosensation. What Alireza found is that the homologue of piezo in plants is actually expressed at the root tips, exactly where this sensation is going to happen. And what he found, I'm not showing you the data, is that if you don't have piezo in plants, their ability to recognize and penetrate hard surfaces is ablated. I show this slide because I find it fascinating that throughout evolution, the same protein has been used for us to sense touch and for plants to sense their surface tension. So take-home messages from my talk is that we've identified sensors of temperature and pressure, And these are essential for many aspects of somatosensation, but also we end up finding unexpected roles in physiological processes. One example I didn't give you is that there's a mutation in PIEZO1 that's actually protective against malaria. So that's something that I never thought of that mechanosensation will take us to. And the other important message is that basic research labs are really the nurseries for future medicines. And I gave you this example, TRPA1, this wasabi sensor that we identified in 2003, almost 20 years later now, is still in clinical trials, and hopefully in the next year there'll be some positive results. But it takes a long time to translate this research into medicines. I end by the names of all current alumni, lab members, and collaborators from around the world, and very importantly, funding on the lower left. But I think if there's a few more minutes, I thought I would end by showing you some fun slides. Some of you might know I'm a little bit addicted to Twitter, and I thought that this Nobel has been such an incredible positive experience, and I wanted to share with you the joy of this scientific discovery and science support through these tweets. But this is a great time for me to remember that I should also tweet this, so I'll take a picture of you guys. And you guys look so awesome. So this was the day that the Nobel was announced, and it's a unique day, especially if you live in the United States, west coast of United States. They announce who's going to get it, our time, 2 a.m. So I had do not disturb on my phone, just like any sane individual would. And so Stockholm called 3 times, but I didn't get it. And then they, I guess, Googled Patapoutian, California, and they found the number, and they called my 94-year-old father, who was awake at 2 a.m. It's another story. First he yelled at them, and then he realized what was going on, and gave them, he called me because he can overcome the do not disturb. So it was kind of a crazy moment where we woke up, found out about this, and they were about to announce it, so we were watching on the computer, the same computer as this. And Nancy, my wife, is the photographer here. She woke up my son, and I love this photo for many reasons. One of them is that you can see how my son Luca is affectionately touching me, which doesn't happen too often. So, and I study sense of touch, so it's a very, very special moment. But one of the things that I've wanted to make sure of is that life doesn't change too much when you win something like this. And the day that we heard, this is October 4 last year, was actually Nancy's birthday, so I had to make her a cake. But Nancy is the actually head of a committee that wants to make sure that my head doesn't get too big. And so she tweets things like this, that I still have to take the garbage out. But winning the Nobel does have a lot of perks. You get a special parking spot that's yours all the time. But I guess one of the biggest highlights for me has been, I'm the first Armenian who won the Nobel Prize, and last June I visited Armenia, and they were so nice. Everybody was so excited. Even the government issued a stamp with my face on it. I'm a bit embarrassed to say, though, that the stamps don't seem to work, because I mailed 25 postcards with this stamp on it, and none of them made it back to the United States. But still, looks good. And my final slide, of course, one of the highlights is you go to Stockholm, which for us, my celebration was over a year, because we initially had a small ceremony in Irvine because of COVID, but this last December, just 2 months ago, Nobel laureates from all 3 years that have missed out all went to Stockholm and received the prize, and you get to really dress up. And here's Nancy, Luca, and I all dressed up, and we had a wonderful time in Stockholm. But anyways, thank you so much for your attention. So happy to be here.
Muchísimas gracias al doctor. En este momento vamos a dar paso a lo ofrecido. Vamos a tener la oportunidad de hacer unas preguntas al doctor Ardem, y para lo cual invito al decano de investigación de la Universidad de San Francisco, César Zambrano. Si alguien todavía tiene interés en anotar una pregunta, por favor levante la mano, que Daniela va a estar por ahí ayudándoles. Seamos muy concisos, por favor, con las preguntas y únicamente referentes al tema. Gracias.
Gracias, Alex. Thank you very much, Professor Ardem. It's a privilege to have you here, and we really enjoy your talk. Before we get to public questions, and if I may, I have a question for you. You mentioned the therapeutic agents for the PIEZO 2, and also several molecules that were being used that were active, meaning antagonist or agonist for the TRPA1 receptor. My question is, how do you design new therapeutic agents nowadays? Do you still use computational methods like QSPR, docking, things like that, or there are new things that you're studying nowadays?
Yes, I should mention that for TRPA1, these molecules are already made and are being tested in the clinic. PIEZO2, we're still looking for these molecules. And the number one way we still look at it is high throughput screening, which means pharmaceutical companies have libraries of compounds, 2 million compounds, and you design this, because they let calcium in, you have a calcium assay where in a 1536 well plate, you can just screen all these million compounds and see the ones that can activate or block the channel. But what you also mentioned with the computational, we're also doing that where, people, if you have the structure, molecules get computationally docked, and then instead of screening 2 million, you go and only screen a few hundred that already have a good chance of blocking or binding to a pocket, which makes things a little bit easier.
Thank you. Diego has a question.
Sorry about jumping, but in the case of temperature, we know that there's variations among different groups as far as they feel temperature. I think the Inuits feel temperature different than the people in the Andes and all that. Do you have the same thing with the case of pressure? Do you have you studied variation among different humans as far as how these receptors work?
It's very difficult to answer because there's so many different processes that involve this. First of all, your just internal state decides sometimes how much you're paying attention. You can imagine you'd be shocked if something touches you if you're not expecting it, but then if you are, it's a very different sensation. And a lot of this is also how you grew up, especially for temperature. It's many times not genetic, but the environment that you live in for a long time, you get adapted to it. But there's also, I'm sure, a strong genetic component. Male and female prefer different temperatures, that's known. Lots of information is about that. We haven't done too much on this, on the human variation, but it's a very interesting concept.
Dr. Ardem, another question. Is there a relationship between the piezos and the emotional regulation, such as the case when a mother touches a son that has fallen to make him calm down?
So this is a very active research from my lab and my collaborators. Again, most of the work we do is in animal models, but also since we have these individuals who are piezo 2 deficient, we can actually look at this. And one of the things that it's coming through, that actually it is true, that many of these affective, pleasurable touch is through piezo 2 as well.
Thank you. Another question, the deficiency of the piezo 2, is it affected by hereditary factors or epigenetic factors?
We have not looked at epigenetic factors on piezo 2. Hereditary factors, absolutely. In addition to the loss of function, we know that there are what we call gain of function, which means that there's too much activity of the protein, and this causes different conditions and syndromes. But whether there's slight differences in piezo 2 activity that makes you a better basketball player or not, we don't know.
This is a quite extraordinary question. Benzodiazepines and barbiturates, used as first responders for anxiety in many places, could research with piezo help find new ways to regulate HPA axis, hypothalamic pituitary adrenalinase, finding new pharmacological solutions without the addiction component?
I'm not so sure. I think they're at some point connected, these two pathways in the brain, but I'm not sure there's a direct effect of piezo to what they're talking about.
Thank you. How do you explain the pain in the person who doesn't feel it?
How do you explain pain in a bit? Well, so we scientists actually use a very different vocabulary. So we think of pain as an emotion. So if you can't feel pain or emotion, then you're not feeling pain. I think maybe what the question was trying to ask is, we distinguish nociception from pain. So nociception is the act of transduction of a painful stimuli into an electrical signal. So that's through the receptors I'm talking about. But pain is a feeling that happens in the brain that is based on this information, but it's a feeling. So I don't know if that makes it clear or make it worse.
Some people mentions pain is real.
Well,
so it's not imaginary.
Pain is as real as any other emotion, but all your emotions are real.
Thank you. How similar are the channels in terms of resistance to the action of tetrodotoxin with respect to TRPM and TRPA1?
I was actually going to add one more answer to the previous question before I answer this, and we were talking about this at dinner last night. There's something called the phantom limb pain. So these are individuals who have lost their arm but still feel pain in their fingertips. It's a beautiful example of this dichotomy. Anyways, we can go on and on about it, but it's an interesting example. I think the TTX inhibition that this person is asking about is something that blocks sodium channels, which are what we call downstream of the sensors. So as I told you, these ion channels transduce physical force into an electrical activity, but that's all they do. There are steps after that which then have to propagate this signal to the brain, and that compound blocks the channels that do that propagation. So they're one after another, but tetrodotoxin doesn't affect the receptors that I talked about directly.
Perfect. I think we have one more question. Well, not. Does haptic feedback in technology interacts directly with piezos? Is this why we can create the sensation of different textures and objects?
This is a question that I'm interested in. The person's asking haptics, whether in virtual reality video games or for people who've lost limbs, can give you the sensation of touch using haptics, electronics. And I'm actually collaborating with a materials scientist, and his take on this is that haptics is very backwards in the sense that all they use is vibrations for you to have the sense of touch. But in reality, our sense of touch is very rich. It senses vibration, it senses your hair moving one way or the other, it senses pressure. So he wants to actually learn from PIEZO and our sense of touch to design novel haptic ways to give us a more rich sensation of touch other than just vibration.
Splendid. This is the last question I think is very relevant to young researchers. In your experience, which academic life advice could you give to a junior investigator?
It's difficult to say it in a very short time, but for me, what has really worked is, I kind of mentioned this, is having the curiosity and doing what you love. I never worried about winning prizes or achieving this and that. It was mainly just the love of science and pursuing the direction and see where it takes me. I didn't come in with a big plan of accomplishing this. It was just the love of doing what you want to do. And so if I have one advice is find your passion and pursue it to its limit.
Thank you very much.